the human brain is one of the most complicated and complex systems in the world in general. It is often compared to the universe, to the whole universe in terms of complexity. And today we've heard several lectures which were devoted to gray matter, black holes, gravity, which are very important for general understanding of the universe, but I suggest you to switch to the other universe, our brain. It is, in my opinion, very important to study brain, since our brain is our personal computer which controls all systems in our body. And what is most important, at least for me, that our identity and personality is also for sure reside in our brain. And maybe unfortunately, researchers at this moment are not so advanced to give you the answer to all questions, but certainly we have a tremendous development of the area of brain research, which is maybe should be called neuroscience. This field in generally accepted it should be called neuroscience. And definitely in my lecture I will initially give you some ideas about how complex the brain is, at what stage the humankind is now in general understanding, in its attempts to understand how the brain functions. And initially, I will tell a bit about limitations which we have when we study the brain. After that, the main approaches which are used now in the field of neuroscience to study the brain. The most interesting maybe examples of results, and after that we will go deeper inside, and I will show you how complicated the brain is at the level of microstructure. And at the end I will exemplify new, let's say, approaches which allow us not only to study brain but to control the brain. Definitely, I think that all of us participate in studies of brain. When we thinking, when we communicating to other people, we are actually sending some messages to other brains, which are big black boxes, and receive some responses. But of course, it is quite interesting to go to see inside these black boxes, to understand, to realize how these black boxes works. And I think that the substantial progress in this research started about a bit more than 100 years ago, when the famous Spanish scientist, who is actually a Nobel Prize winner, Ramon y Cajal, found that our brain is not, how to say, the entity, but it contains actually a separate small units, and these units are now called neurons or glial cells. And from this time, I think that the current neuroscience research is really started. You can see Ramon y Cajal it is by his microscope. I would like to attract your attention that 100 years ago, microscopes looks like this one. And what he found is shown here. This is an original drawing by the hand of Ramon y Cajal, and we can see the cells, which is now called the neurons. We can see that they are quite different in terms of their morphology. I think that you can recognize several different types. And at that time, as I said, neuroscience was started. And for the recent, let's say, 100 years, we found many interesting but very complicated things. It turned out that our brain consists of about 80 billions of neurons, like neurons which you could see on the previous slide. And of course, all these neurons participate in computation in our brain, and in order to completely understand the brain, we need in some way to see where these neurons are located, what are differences between different neurons, how they connect to each other, and how they communicate to each other. And if we, how to say, take into account that this is about 8 billion of entities, it means that the task which we should solve is a very complicated one. But on the top of, let's say, just general organization of the brain and connectivity between different neurons, it is quite important to know that neurons process the information by some special electrical signals, which are called action potential. You can see the example of such electrical pulse, let's say, and one neuron communicates to the other neuron using this electrical signal. Actually, as you understand, electrical signals cannot be visible, but scientists solved this problem, and now they are visible. And on the top of, let's say, just general connectivity, we need to know how the brain processes a particular information by exchanging of these electrical signals between different cells in the brain. To do that, the researchers developed a new types of microscopes. I would like you to compare this microscope to one which you saw about two slides ago, and you can see that it is tremendously different apparatus, and actually this is just an optical part of the current microscope, because we cannot see here lasers and different light recording devices and computers which are necessary in order for such system to work. But in spite of, let's say, very substantial progress in the method of recordings, we still have several problems which are specifically related to studies of brain. As maybe you know from school, we are mainly composed of water. About 80% of our body, and the same are for brain and neurons, is composed of water. And extracellular medium is also water. And when we look at the microscope, we saw water in water. Of course, in this case, you don't have any contrast, and if you use just ordinary microscopy, you will see figures, photos like this one. Yeah, there is something. Definitely, these are different cells, but no specific details cannot be observed. Actually, we know now that except of water, we are composed of proteins. This is the most substantial part of our brain and neurons inside our brain. And proteins are just small molecules which are impossible to observe using microscopy, transmitted light microscopy. And this is the second problem. And the third one is that in spite of being composed of water, we are not transparent. If I put my hand, you cannot see my eye, and I cannot see the audience. And this is just about 1 centimeter of my flesh, let's say. But we need to see inside the brain, and definitely you can see I have a skull, and most mammals have skull, and it is a problem how to see actually cells inside the brain. And to cope with this problem, there was this method, the relatively new method was invented, which is based on so-called fluorescent microscopy. The fluorescent microscopy is also relatively new field in optics. Let's say it is about 100 years old, but actually the main idea behind the fluorescent microscopy and its application to biological field is that small fluorescent molecules like this one, they are called fluorescent dyes, they can emit light. And this light is so bright that the current microscopes can record this light. And if you have such dyes, and we manage to attach this dye to the protein under study, in this case, we can see the place where the protein is located. We can choose dyes of different colors, and in this respect, we can choose, let's say, we can follow the distribution of different proteins, and we know that some proteins are specific for some neurons, and in this respect, we can pinpoint different types or different classes of neurons in the brain. Moreover, it is quite important that literally recently, about 10 years ago, some fluorescent dyes were modified in a way that can change the conformation. These are actually molecules. And when the conformation is changed, these changes can take place during electrical activity of the neurons. And changes in the brightness fluorescent molecules will allow us to see if neurons are actually electrically communicating to each other. Just to give you a proof of concept, I would like to show you this glass of water. Actually, you cannot see water. You see only some marks on the other side of the glass. But if you place just a small drop of fluorescent dye in this water, you see a tremendous, interesting picture of, let's say, some vortex inside this water. And this vortex is visible only in the place, in the sites where molecules of the dye is distributed. Besides, of course, we cannot, how to say, drill our skull and to study the brain of human beings, and therefore we are restricted to other animals, and it turned out that some small animals are actually transparent. Some worms, some larvae are actually transparent. And if we use this type of animals, in this case, we don't need anything, we don't need to drill anything, and we can use fluorescent approaches together with usage of these small animals. And this is an example of such an animal. This is a larva of a small aquarium fish, which is called zebrafish. I think that many of you have such a fish in your aquarium. But the larva of this zebrafish is very, very small. As you can see, it is just about, this is a 0.1 millimeter. It is about a half of millimeter, the head of this animal. And the dots, the bright dots which you can see, are actually the separate neurons within the brain of this animal. And you have, now we are observing 3 projections of this head from the top, from the side, and from the front. And I will show you how this cells works during the normal activity of this animal. This animal has relatively small, it's not brain actually, it is distributed network of neurons, and it contains about 75,000 neurons, which is enough for this creature to do a very complex patterns of behavior. As you can see, there is a general activity, and when you observe some very, very powerful flashes, some food actually is presented to this animal, and we see how it results in very substantial excitation of the whole network. Certainly we can we can demonstrate to this animal different types of things, or to produce some different action on their sensory system, and as a result we will see some different patterns of activity of the neurons, and I think that in a due course researchers will be able to pinpoint and to understand how, let's say, this activity actually is realized in computation. About 10-15 years ago, the same could be done with much more complicated animals. These are pups, these are mouse pups, and just to show you proof of concept, in each cell of the body of this pups, of these 3 green pups, some green fluorescent dye is expressed, and we can see each cell of the body of this animal. Actually, these pups and mice in general are very developed creatures compared to the previous one. Their nervous system, they have, of course, brain, and their brain contains about 75 million of neurons, which is 1,000 times more than in small zebrafish. And of course, it is a very complicated system, which is relatively close to our brain. And at this moment, mice and a bit, and maybe rats are considered as a well-developed model for studying the properties of our brain. And I will show you an example of results from our laboratory. This is a part of the spinal cord of mice, in which fluorescent dyes, which can respond to electrical stimulation of neurons, are expressed in different colors in two different types of neurons. As you can see, there are green and red, let's say, neurons, and there is the activity. You can see the changes in fluorescent intensity in these neurons, and actually this are recording which is done when a painful stimuli applied to the periphery of the animal, and actually it should result in expression of emotion of pain in the brain of this animal. And it is, let's say, spinal cord. It is intermediate location in the brain of this, actually in the nervous system of this animal. And we try to understand how information about peripheral pain is encoded in the spinal cord during the processing in the nervous system. However, as you could see on the previous slide, we could observe just, let's say, several neurons and many processes of the neurons. But using very, very advanced approaches, this is a result of which was published just recently. It was possible to record activity of 10,000 neurons in the cortex of mouse. And this is a relatively large region. As you can see here, it is about 4 millimeters by 4 millimeters and about 200 microns deep inside the brain. And actually, it allows us to simultaneously record this activity. And you can see the changes in activity as white dots, which are on somas of some of the neurons which we observe in here. And basically not just fluorescent methods used for this, but it is possible to use direct electrical recordings by very advanced semiconductor systems. But unfortunately, it is also possible to record from several thousand of neurons. And this is, how to say, our limit at this moment. Several thousand, up to 10,000 neurons. If we are talking about a human being, which has about 80 billions of neurons, it means that we are at the beginning of our way to understanding how our brain works. But if we are talking about simple organisms, or like larva of zebrafish, which has 75,000 of neurons, it means that it is very close to solving the problem, and I hope that maybe all of us in, let's say, in 10 years period will know how the brain, how the network of neurons in very simple organisms actually compute. It is very interesting for solving several problems, practical problems. We need to know how our brain works, and we hope that the ideas which we will find in these small creatures will also apply to processing of information in our brain. And second, I think that it is possible to create a new type of computers which are based on, let's say, now we can grow neurons separately from the body, our neurons or neurons of mice or rats. And after that, I hope that it will be possible to establish specific communications between these neurons, and these neurons will solve a lot of computational problems which are now solved by our semiconductor-based devices. Okay, but situation is much more complicated since neurons themselves are not just their bodies. You can see their bodies here, but basically they have a lot of protrusions which are called dendrites, and you can see these dendrites, and now we know that information processing has mainly place in direct contacts between these protrusions. And each neuron establishes about several thousand connections with other neurons, which means that altogether we have more than 100 trillions of contacts between the cells, between the neurons in our brain. And definitely to understand how the brain works, we need to know everything about these connections as well. Now we study these connections separately. We try to understand how a single connection works. But after that, we will hope, to apply this, the discovery related to work of single contact, which is called synapse, to the whole brain. Okay. This is an additional demonstration of complexity of our nervous system. This is a dendrite which you could see on the previous movie, and this is a very, very small portion of the brain which was reconstructed in very details. This is about 15 micrometers long and 7 micrometers in diameters, and here we have 800 of elements of other cells which are located in this area. It means that, interconnections between our neurons is tremendously rich. And I hope that young people who are in this audience now will solve the problem why we need so complex system of connections between our neurons. Okay, the other way how to observe the whole brain is a method which is called clarity, which was developed about 10 years ago, and it solved actually the problem about which I told you. Our tissues are not transparent, but applying specific chemical processing of the tissue, we take a specific type of the molecules which are called lipids, and which give our which make our brain and brain of animals to be transparent. And you can see the results of this processing. This is a mouse brain, and this is also a mouse brain, but after processing with this clarity approach. As you can see, I hope you cannot see the brain, but brain is here, and it is completely preserved. And as a result, we can use light microscopy to reconstruct the whole system of the neuron connection in the mouse brain. This task is now solved at the level, let's say, of cell soma and dendrites. But of course, we cannot, this approach does not allow to see each connection between each neuron. Here is, let's say, fantastic, in my opinion, demonstration of how this upper part, this is a cortex of mouse brain. Below is hippocampus, which is quite important for creating memory in the brain of mice and in brain of people. And this is how this method works. We can see each neurons with very, very tiny details. We can load different fluorescent dyes in different types of neurons. And this is a hippocampus, and a lot of different type of cells are presented here, and we can observe these cells. Okay, but also just maybe about a couple of slides about the other ideas. Okay, everything which I told you about was related to how we can see different parts of brains, how we can understand the connectivity between the neurons, and maybe how to see the functioning of the neurons. But of course, for the practical purposes and in order to validate our discoveries, we would like to control nervous system. It was done, let's say, maybe for 100 years using some types of electrical metal electrodes which can be inserted in the brain, and it was possible even to do with human brain in certain situations. But with development of new approach, which is called optogenetics, and which is based on a very simple idea, which all of us observed maybe in our own life, but we didn't pay an attention to this. This is, let's say, bacteria which are single-cell organisms. But if you try to apply light to this, if you would like to apply light to this bacteria, they start to move out of the light. It means they don't have eyes, they don't have legs, but in some mysterious way they feel light and they go out of the light. And it was realized that a specific protein which is expressed in this bacteria is actually very interesting. It is molecule, and it is photosensitive, electrically coupling molecule. It can absorb a quantity of light, and after that it can be converted in a path for the electrical current. And if we, using the current approaches, if we insert the gene of this protein to neurons of our or animal body, in this case it is possible to activate these neurons just by flashes of light. And I will show you an example how we control a movement of actually locomotion of mice. This is transgenic, so-called transgenic mice, in which this protein is expressed in the cortex, in the specific part of the cortex, which actually is responsible for starting of locomotion. And at the beginning, you can see that there is, let's say, there is a light guide which is inserted to the cortex of this mice, and it was very silent at the beginning. But after that, when light was switched on, it started to move around. And immediately when the light, you can see this blue light, yes? And immediately when light is stopped, the animal will be also stopped, you see? No, it is, how to say, it is very relatively old movie. It was done by about 10 years ago. Now it is possible to control many different functions using the light. It is possible to activate neurons, inhibit neurons, or control many biochemical processes inside our cells using lights of different colors. And I hope this research has a very good perspectives as a way of treatment of many diseases, and in particular, it is, how to say, it is always amazing to work with this approach because of, let's say, you can see on this photo, this is a photo of young investigator Ivan Blashak from our laboratory who actually does this research, and you see it is fascinating to do this research. And actually, at the end of my talk, I would like to conclude with some optimistic statement that I hope that we have so good brains that it will allow us to finally understand how our brains work. And to, how to say, as this slide states, you need to have good brains to study brain. And I will pass the other part of my lecture, of our lecture, to Nana Voitenko, who in particular will explain you how to use your brain on the good, how to keep your brain in good shape in order to understand how brain works.
Thank you. Yeah, thank you, Pavel, for such a nice videos. I also was amazed. Yeah. And now let us figure out how we can use it, because it's a beautiful picture, but what is practical meaning of this? So as Pavel said, the very simple base of the functioning of our very complicated brain. So each neurons receive signals and pass signals, and this is the main idea of existence of every neurons, to receive signal and to pass signal. And where the signals come to the neurons, for example, to peripheral neurons, the signals come from outside of our body through the sensitive organs of the body, like vision, hearing, smell, taste, and touch. So all this incoming signal could produce this electrical current, which we called action potentials. And each neuron has connections to, as Pasha said, thousands of other neurons. And since we have about 100 billion of neurons in the brain, which make about 100 trillions of contacts, of course, we can see this network which allowed us to operate by and control all our body. And exactly the number of these connections is reflect the level of our brain development, the level of our intelligence. The more connections, the higher level of intelligence. And how to increase the number of these connections. So let's start to the movie. You see, this the place of the contact, we call it synapse, is activated only then the signal arrives to this place. So we can activate synapses, the contact, only in activity-dependent manner. So we need to send the action potential to there. So that means that we need to receive the quant of information. And all this number of these contacts, of course, it's true for adult brain. But when the just newborn baby came to the life, of course, the baby has much less connections in their brain. Of course, newborn baby can receive the information, it can feel all incoming signals, but in terms of intelligence, the brain of the newborn baby is tabula rasa. It's like a blank field on which we can write down the information to activate as much connections as possible. And that is why it is quite important to study during the childhood. More information, more contacts. That is why it's quite important. Really, today, all kids who visited all this event during this science festival really increased the number of their contacts by, I don't know, 100,000 new connections they received during this event. Here you can see it's artificial colors, but violet is a more mature brain, and you can see that then the child is just 3 or 4 years old. His or her brain is not matured enough, but you see how the brain is changing during the development. That means that more information, more connections, and finally, 90% of all possible connections is finalized at 21 years old. So, and after that, during whole your life, you can use it. And of course, you can ask me, so nothing would happen after 21 years old? No. The good news is you can use it and you can keep all the connections that were established during your childhood. And how to keep this level of connectivity in our brain? Because you know, the new contact established when the signal arrived, but if you do not use this contact, it can degrade. So that is why during whole your life, you should pump your brain like we do in gym. So in gym we train our muscles, but also we can train our brain by reading, by studying new languages, playing chess, playing different musical instruments. So all this allowed us to keep our connections in active states. And of course, if we will, during all our life, keep our connections active, so it will prevent, for example, the development of dementia. And also you can ask me, maybe there is some magic pills or techniques which would keep all these connections active. I just take a pills, or I don't know, put some helmet on my hand, and so I will be genius or expert in any field. So the answer is no. No, it's not possible. Of course, there are a lot of techniques which could increase your memory, improve your memory via some association, a lot of methodics. But all these techniques require work, require efforts. So nothing magic, just to keep your contacts active. This is the magic. And you can do it, but our brain is very lazy. It's never want to work, never want to analyze information, receive information, because it requires a lot of energy. At the peak of their activity, brains can use up to 70% of energy which is in our body. And of course, brain does not want to waste the energy, but we should somehow push our brain to work, to analyze, to memorize, to do something what we need to keep our brain in function mode. And also, not only the study, also the physical activity would help us to keep our brain in a good mode, because it also activates different connectivity in the mosque. And I can tell you about a recent experiment which was done in one American university to study senile dementia. They create two groups of senior people, over 80 years old in each. And one group go to the gym 3 times a week for quite simple exercises, and another group does not do that. In 6 months, that group who worked out in gym increased their ability to solve arithmetic tasks by 50%. Can you imagine? 80 years old. So they are all immediately have to go to gym to improve our activity of our brain. And it is important, independent of our age, to have physical activity like walking, hiking, traveling, biking, whatever we can. And also, of course, the good food, because our brain needs some building materials to rebuild ourselves, to rebuild all this mechanism we were, the Pasha will talk us about. And especially also important vitamins and trace elements, like, for example, with vitamin D. The vitamin D is now quite a lot of study that shows studies show the correlation between deficit of vitamin D and development of neurodegenerative diseases like multiple sclerosis or even Alzheimer. So in Africa, there is no multiple sclerosis at all. And in Scandinavian country, it's quite common disease because lack of sun, because evolutionary, nature would not somehow suggest that the people would be covered by clothes without sun. So we need to somehow to have this vitamin D to keep, first of all, our brain in order. Healthy sun. This is my lovely part of my talk. So you know that the average person should sleep 8 hours a day, but sometimes it's very individual. Maybe 7 hours, maybe 9 hours. It depends upon the age. So the kids need more to sleep, and older people less. It's individual, depends upon age, health, and genetical background. But everyone needs to sleep. Why? Why we need to sleep? Why our brain needs to sleep? I would tell you that our brain never sleeps. Never sleeps. It's always, it always works to keep all our function, but it switches its activity slightly. For example, the sensory activity is switched to minimal mode, but during the night, during the sleep, our brain modifies neuronal network. And first of all, it has to our memory. Where is our memory? You already hear the word hippocampus. Scientists know that our memory is, first of all, in our hippocampus. Hippocampus is such a gyrus inside our brain which reminds the seahorse by form, that is called hippocampus. And the information we received first stored for a while in hippocampus. It reminds me random access memory in the computer. And in some time, in a while, this information is transported to the cortex for long storage, long-term storage. And here you can see the transition of our memory from hippocampus to cortex. And this process takes place only during sleep. During sleep, we somehow pass all our information to cortex, and after we wake up, our hippocampus is like a formatted disk ready to accept the new information. And studying the influence, studying this process of transmission of the memory from hippocampus to cortex, scientists also noted that this process depends upon the emotion. So, and you know that the emotionally colored things, it's much better to remember, like first love, or lyrics with the music which we like. It's much more easy to remember lyrics with music than without music. And you can use it on practice. To remember something, you just have to tie this to some pleasant emotion. It's better to remember. And also, the process of this memory transformation or consolidation took place during the sleep. And to remember something, we need to sleep. And we not only have to sleep, like for 15 minutes, we need to have the healthy, long-lasting sleep, because it's important to have deep sleep, and also here the new, quite recent publication which also tell us about the importance of so-called rapid eye movement sleep. So the whole these stages of sleep is important to remember for a long period of time something. And also during the sleep, during this process, it's like we fulfill the puzzle of our worldview. Even you maybe sometimes noted that something which you try every time to put, you see, pieces of puzzle in place, but sometimes during your sleep, in a dream, the pieces of puzzle, takes the right places. You remember the Einstein theory of relativity, or René Descartes' Tractate, scientific method, or Mendeleev periodic table. All this came to them in a dream, because they work hard, they think a lot, and due to consolidation, the puzzle was done. And due to this consolidation, in every language we have proverbs like the morning is wiser than evening, yeah, or you can sleep on the problem. This is because of consolidation. Sometimes if you think about something before you go to sleep, in the morning you will have the right decision. And so the last thing I would like to tell you about today is, you know that we have in our body so-called lymphatic system, which help our body to clean out of some metabolic products, toxins, whatever, any garbage from our body. But for some reason, this system is not in our brain. Why? Just recently, Danish scientist Maiken Nedergaard discovered such a system, cleansing system in the brain. She called it a lymphatic system because using the glial cells, this allowed us to get rid of all garbage from the brain, including misfolded proteins, so-called beta amyloid. So, and this system works only during sleep. This experiment shows us that only during the sleep, you see here, this system works and help us to get rid of all garbage from our brain. And you understand that beta amyloid, if we sleep less, or we have lack of sleep during the long period of our life, it would lead to the increasing amount of beta amyloid in our brain, which could also produce the Alzheimer or other neurodegenerative disease. So it's very important to sleep and to clean our brain every day from all the garbage. And thank you for your attention. Take care about your brain.